Pooja,
Yes.
ma'am, sorry.
Yeah. Yes, Amina.
Sorry, ma'am. I thought that someone,
It's okay.
any
It's okay.
friend of mine.
It's okay.
Hello.
Am I audible?
Yes, ma'am.
Okay, so I'll start sharing my screen and we'll start with our session. Is my screen visible?
Yes, ma'am.
Uh, are my slides clear? Okay. Okay, so we'll start with our session. So today we'll start with our problem solving session for week 2, and the lectures, the questions from that we will be covering are from lecture 6 and 7. So last week we have completed till lecture 5. Today we will be completing lecture 6 and 7, which includes cellular metabolism part 1 and part 2, lecture 8 and 9, cell cycle and control part 1 and part 2, and lecture 10, program cell death. So this course, molecular biology, is being taken by Professor Vishal Trivedi. He is professor in IIT Guwahati in Department of Biosciences and Bioengineering. I'll briefly introduce myself as well. I am Pooja Yadav. I am a PhD scholar and a PMRF fellow in Department of Biosciences and Bioengineering in IIT Bombay. So I am a PhD student in molecular virology lab. Like previous session, so we'll have questions, and uh, first I would expect you guys try to answer the question, and then we will look into the concept for each questions. But first let's try to give answer from your side first, and then we'll look into the concept for each of the question. So let's begin with our today's session. um, is there any question or a doubt? Hello. Okay, so let's begin with our today's session. Um, question number one. Question number one states that in eukaryotes, the enzyme for glycolysis and the Krebs cycle are found in option 1 cytosol and Golgi apparatus respectively. Second option is inner mitochondrial membrane and endoplasmic reticulum respectively. Third is mitochondrial matrix and cytosol respectively. Fourth is cytosol and mitochondrial matrix respectively. You can either unmute yourself and or you can write the answer in the chat box. We have one answer, option number 4, cytosol and mitochondrial matrix. We have fourth as an answer in the chat box. Is there any other option that might be correct for this question apart from option 4? Okay, so option 4th is the correct option. So glycolysis, it occurs in cytoplasm, and Krebs cycle, it occurs in mitochondrial matrix. So just briefly look into what is glycolysis and Krebs cycle. So basically, the glycolysis is the first step in the cellular respiration, and it occurs in the cytosol of the cell. So it is a universal metabolic pathway that breaks down the glucose into pyruvate. So glucose is a 6 carbon molecule, while the pyruvate is a 3 carbon molecule. So one glucose molecule, it forms two pyruvate molecule. So this glycolysis process does not require oxygen, so it is anaerobic process. So basically, we need ATP for we need energy, which is ATP. So to finally get an ATP, the first step of ATP production is the glycolysis, where our complex carbohydrates are break down into glucose, and glucose then goes into glycolysis pathway, and it forms pyruvate. Further, the formed pyruvate, it goes to Krebs cycle. So Krebs cycle, it occurs in the mitochondrial matrix, and it is the second stage of cellular respiration. It requires oxygen, so it is an aerobic process, and the complete oxidation of glucose by breaking down pyruvate into carbon dioxide. So this cycle generate high energy electron carriers NADH and FADH2 and ATP. So from glycolysis, we get ATP and NADH. In from the Krebs cycle, we get ATP, FADH2 and NADH. So further, these NADH and FADH2 goes to electron transport chain. From here we get ATP from these NADH and FADH2. So finally, we need to get our energy in the form of ATP. Either these pathway, either they produce direct ATP or they produce ATP indirectly by producing either NADH or FADH2, but ultimately we require energy in the form of ATP. So finally, these NADH and FADH2, they goes to electron transport chain, and where we get our final ATP from these two products. So the correct option for this question would be option number fourth, cytosol and mitochondrial matrix respectively. I hope this question is clear. So if you guys have any doubt, you guys can unmute yourself or write in the chat box about your question. So okay, so if this question is clear, let's move to our next question, question number 2. Question number 2 states that the precise biochemical role of hexokinase in committing glucose to the glycolytic pathway. First, it establish a metabolic barrier preventing glucose from entering glycolysis. Second, it forms a stable enzymatic complex with glucose, ensuring metabolic integrity. Third, it initiates a cascade of biochemical reaction, irreversibly dictating glucose to glycolysis. Fourth, it finally regulates ATP production. Please try to answer this question as well. We have one answer, option number 2. try to remember the role of the step in which the hexokinase is involved, and then try to answer this question. Is there any other option that is correct apart from option number 2? So the correct option for this question is option number 3. It initiates a cascade of biochemical reaction, irreversibly dictating glucose to glycolysis. Let's see how the option number 3 is correct and option number 2 is not correct. So hexokinase, it is involved in the first step of glycolysis, where it converts glucose to glucose 6 phosphate. So it is by for by releasing one phosphate from ATP, which gets bind to the glucose, which forms glucose 6 phosphate. So hexokinase, it is a first enzyme in the glycolytic pathway, and it plays a critical role in converting glucose to glycolysis. So basically how it works is phosphorylation of glucose. So hexokinase, it catalyzes the transfer of phosphate from ATP to glucose, forming glucose 6 phosphate. So this reaction is irreversible under physiological condition, ensuring that the glucose which is trapped inside the cell is continue to the glycolysis process and ultimately form the pyruvate. So so in the second option, what is given, it forms a stable enzymatic complex, which here it does not happen. Hexokinase does not form any complex with the glucose. It uh, it catalyzes the transfer of phosphate from ATP to glucose, which leads to the formation of glucose 6 phosphate. And this reaction from glucose to glucose 6 phosphate, catalyzed by hexokinase, is an irreversible reaction. Once the glucose gets converted to glucose 6 phosphate, it leads to the formation of pyruvate by multiple steps. So this reaction is irreversible reaction. So once it gets catalyzed, so further the 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 upcoming reaction they take place, and which ultimately lead to the formation of pyruvate from glucose. So once glucose is phosphorylated, it cannot leave the cell or be used for other metabolic process. So unless it is dephosphorylated. So this step effectively commits glucose to glycolytic pathway. So the correct option for this question is, it initiates a cascade of biochemical reaction, irreversibly dictating glucose to glycolysis. I hope this question is clear to everyone.
Ma'am, if hexokinase doesn't leave that particular reaction, then how it is performing a cascade of biochemical reaction? Is it for glycolysis?
It is just initiating the reaction. It is just initiating. It is the first step of the glycolysis. So that is why it is initiating a cascade of biochemical reaction. It does not catalyze all of the reaction, but it initiates main reaction, which is the glucose to glucose 6 phosphate. So it works as the initiation of glycolytic pathway.
And it is said that cascade means in that glycolysis pathway only, due to its catalysis, the whole reaction is performing. That is why it is said that it is a cascade of
So
biochemical reaction.
basically, this reaction is irreversible reaction. Once the glucose is converted to glucose 6 phosphate, so it has to go to multiple reaction to form a pyruvate. So it is working as the initiator of glycolysis. So ultimately, what it is doing, it is initiating a one reaction which is continuing the cascade of multiple reaction.
Okay.
So I hope this is clear. So if this question is clear, let's move to question number 3. Um, Question number 3 states glycolysis is inhibited when first ATP to ADP ratio is low, second concentration of fructose 2,6 biphosphate is low, third is NADH plus H plus um, ratio to NAD plus ratio is low, fourth is concentration of sugar in the cell is high. So this is basically NADH2 plus, sorry, NADH plus H plus uh, 2 NAD plus ratio is low. Please try to answer this question. So try to remember the regulation of glycolytic pathway. So how the glycolytic pathway is regulated when we have a higher ATP uh, in the cell or other molecules. So there is this regulation of glycolytic pathway occur. So try to remember what happened in the regulation of glycolysis. Okay, so let's see how the regulation of glycolysis occur. So the correct option for this question is option number second. Concentration of fructose 2,6 biphosphate is low. So glycolysis, it is a very regulated, tightly regulated by several factors, including the concentration of fructose 2,6 biphosphate. So which is a potent activator of glycolytic enzyme, um, uh, which is phosphofructose kinase. So here how it works, what is the role of fructose 2,6 biphosphate? uh, Fructose 2,6 biphosphate, it is a signaling molecule that activates this enzyme, which is phosphofructokinase 1. This enzyme is responsible for catalyzing the committed step of glycolysis, which is the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. When fructose 2,6 bisphosphate is higher, the the glycolysis, these glycolysis, it is stimulated, while when it is low, uh, this enzyme is also decreased, so the glycolysis is inhibited. So what fructose 2,6 biphosphate is doing, it is activating this phosphofructokinase enzyme, which is catalyzing the reaction fructose 6 phosphate to fructose 1,6 biphosphate. So ultimately, this this the concentration of 2,6 bisphosphate is regulating one of the regulating factor of uh, the regulation of glycolytic pathway. So the correct option would be concentration of fructose 2,6 bisphosphate. So when ATP to ADP ratio is low, so we need more ATP. So in this case, the glycolysis pathway would is not inhibited. It would occur but not inhibited. While when there is the low concentration of NADH to the ratio of NAD plus, this again we have a lesser number of energy, so we would require more amount of energy. So the glycolysis pathway in this case also would not be inhibited, but it will be more we will be needing more of the glycolysis pathway. While the concentration of sugar in the cell is high, this also does not inhibit the glycolysis pathway. When we have a higher sugar level, the sugar breaks down into the glucose and it leads to the glycolytic pathway. So the correct option would be second, the concentration of fructose 2,6 bisphosphate is low. I hope this question is clear. So if this question is clear, let's move to our next question, question number 4th. In a cellular study, researchers manipulate the availability of oxygen to investigate its role in the Krebs cycle. If oxygen is limited, the impact on overall Krebs cycle activity. Option number 1, increase in ATP production. Second, increase NADH production. 3 is accelerated citrate synthesis. Fourth is inhibition of Krebs cycle. Please try to answer this question. We have one option as an answer, option number 4, inhibition of the Krebs cycle. Is there any other correct option apart from option number 4? So the correct option is option number 4, inhibition of the Krebs cycle, because as we have studied in the question number first, Krebs cycle is aerobic process. It requires oxygen to occur. So when the oxygen is limited, so there will be the inhibition of the Krebs cycle. So if there is an inhibition of the Krebs cycle, it decreases the ATP production. So for this matter, option number 1 is incorrect. It increased the NADH production, which is also incorrect, because during the Krebs cycle we have the synthesis of NADH, and when the Krebs cycle is inhibited we have lower NADH production. Also accelerated citrate synthesis, which is also not true, because citrate is being synthesized in Krebs when Krebs cycle occur. So when there is inhibition of the Krebs cycle, uh, there will be no citrate synthesis. So Krebs cycle is, which also known as citric acid cycle, is an aerobic process, meaning it requires oxygen to function properly. Oxygen is also essential for the regeneration of NAD plus and FAD from NADH2 and FADH2, uh, which are produced during the Krebs cycle. So when we have NADH and FADH2 from the Krebs cycle, it goes to electron transport chain, which ultimately lead to the regeneration of NAD plus and FAD. So in electron transport chain, we have oxygen as our last electron acceptor. So in that matter, oxygen is also important. So what is the role of oxygen in the Krebs cycle is the NADH and FADH2, which are generated in the Krebs cycle, they donate electron to the electron transport chain in the inner mitochondrial membrane. So oxygen act as the final acceptor in the ETC, allowing the regeneration of NAD plus and FAD, which are required for the Krebs cycle to continue. So basically, when these generated NADH and FADH, they goes to electron transport chain with the help of oxygen. We again have the regeneration of NAD plus and FAD, which are required for the continuation of the Krebs cycle. So when there is no oxygen or oxygen is limited, the inhibition of the Krebs cycle would occur. So I hope this question is clear. If there is any doubt, you guys can ask in the chat box, or you guys can unmute yourself and ask here as well. So if this question is clear, let's move to our next question, question number 5. The consequence of high concentration of NAD plus H plus on the Krebs cycle. First, it inhibits citrate synthesis. Second, it inhibits alpha ketoglutarate dehydrogenase. Third is it activates isocitrate dehydrogenase. Fourth is it activates fumarase. Please try to answer this question. We have two answer, option number 2 and option number 3. Is there any other option that might be correct apart from option number 2 and 3? Um, the correct option for this question is option number 2. It inhibits alpha ketoglutarate dehydrogenase. Let's see how this option is correct. So the Krebs cycle is basically very tightly regulated, like the glycolytic pathway or the glycolysis, by the concentration of its substrate and product, including NADH. When we have a high NADH concentration, so the cell has sufficient reducing equivalent uh, or energy, which lead to feedback inhibition of Krebs cycle. So first, this enzyme alpha ketoglutarate dehydrogenase, it is a key regulatory enzyme for the Krebs cycle. So it is strongly inhibited when the concentration or the level of NADH is very high. Sorry. So basically, the when the concentration of NADH is very high, this inhibits this enzyme alpha ketoglutarate, which hinder this catalysis of alpha ketoglutarate to succinyl coenzyme A. So this inhibition slows down the conversion of alpha ketoglutarate to succinyl coenzyme A. So basically reducing the overall activity of the Krebs cycle. So when we have a high concentration of NADH, this enzyme alpha ketoglutarate dehydrogenase, it gets inhibited, so which further inhibit the catalysis of this reaction, which further lead to inhibition of the Krebs cycle. So the correct option for this question is it inhibits the alpha ketoglutarate dehydrogenase. I hope this question is clear. Okay, so if this question is clear, let's move to our next question. Question number 6. The question number 6, it states the primary enzyme responsible for the activation of fatty acids before their entry into beta oxidation is, first option is carnitine palmitoyl transferase 1, second option is acetyl coenzyme A carboxylase, third option is fatty acid synthase, fourth option is acyl coenzyme A synthetase. Please try to answer this question. So before the fatty acid, it goes for beta oxidation, so they has to be activated. So what is the primary enzyme which is responsible for the activation of those fatty acid? So please try to answer this question. We have one option, option number 1. Is there any other correct option for this question apart from option 1? The correct option for this question is question number 1 only. Carnitine palmitoyl transferase 1. So let's see. The correct option for this question is option number 4, acyl coenzyme A synthetase. Let's see how this is correct option. So, this carnitine palmitoyl transferase 1, it is responsible for the transfer of fatty acid to the mitochondria. It is not responsible for the activation of fatty acid. So basically, this before the fatty acid they can enter the beta oxidation pathway, they must be activated. So this activation process, this is fatty acid. So this activation process involves the attachment of coenzyme A molecule to the fatty acid, forming the fatty acyl coenzyme A. So this enzyme, the enzyme which is responsible for the catalysis of uh, fatty acid to fatty acyl coenzyme A is acyl coenzyme A synthetase. This fatty acid, it's not an active form, while this fatty acyl coenzyme A is active form of a fatty acid which goes to mitochondria where the beta oxidation take place. So how this reaction is catalyzed? We have fatty acid, we have coenzyme, we have ATP, and in the presence of ATP, this reaction is catalyzed by the enzyme acyl coenzyme A synthetase, where this thioester bond formation occur between coenzyme A and the fatty acid. So this is how fatty acid it gets activated, where it goes to mitochondria, where further the step of beta oxidation take place, and we have the final acyl coenzyme A and acetyl coenzyme A after beta oxidation. So the correct option for this question is acyl coenzyme A synthetase. I hope this question is clear. Okay, so if this question is clear, let's move to our next question, question number 7. Question number 7, it states in eukaryotes, beta oxidation primarily occurs in first option is Golgi apparatus, second is endoplasmic reticulum, third is mitochondria, fourth is nucleus. Please try to answer this question. We have one option 3. Yes, the correct option for this question is mitochondria. So in eukaryotes, beta oxidation is a process basically by which the fatty acid are broken down to generate acetyl coenzyme A, which can then enter to the Krebs cycle for energy production. The location where the beta oxidation take place is the mitochondrial matrix. Short and the medium chain fatty acid, they can directly enter to the mitochondria, while the long chain fatty acid, they require the carnitine shuttle system to be transported to the mitochondria. So I hope this question is clear. This was a very straightforward question. So the correct option is mitochondria. So let's move to our next question, question number 8. Acyl coenzyme A synthetase catalyze the formation of dash linkage between the carboxyl group of fatty acid and the thiol group of coenzyme A. First option is thioester linkage, second option is amide linkage, third option is phosphoester linkage, fourth option is phosphoanhydride linkage. Please try to answer this question. We have one option. We have two options as an answer, option number 1 and option number 3. Okay, so is there any other correct option apart from option number 1 and 3? The correct option for this question is option number 1, thioester linkage. So, in the first process where the fatty acid has to be activated, so we have uh, um, these acyl coenzyme A and the fatty acid. Um, so fatty acid synthetase, it is the enzyme responsible for activating fatty acid by forming a thioester linkage between the carboxyl group of the fatty acid and the thiol group of the coenzyme A. So this reaction is essential for the fatty acid to enter the metabolic pathway like beta oxidation. So how this is converted? We have fatty acid, we have our coenzyme A. The fatty acid has carboxyl group while the coenzyme A has SH group. So in the presence of ATP, the enzyme acyl coenzyme A synthetase, it forms the thioester bond between the carboxyl group of the fatty acid and the SH group of the coenzyme A. So then it forms the fatty acyl coenzyme A and it releases one phosphate from ATP, which leads to the formation of AM, which sorry the release of two phosphate group N, which leads to the formation of AMP and two phosphate group, two inorganic phosphate groups. So the correct option for this question is option number 1, thioester linkage. I hope this question is clear. So if this is clear, let's move to our next question, question number 9. If a cell lacks functional carnitine acyl transferase 1, first, there is an increase in fatty acid activation. Second, it will inhibit fatty acid transport into mitochondria. Third, it will enhance beta oxidation. Fourth, it will promote fatty acid synthesis. Please try to answer this question. We have one option as an answer, option number 2. Is there any other correct option for this question apart from option number 2? So the correct option for this question is option number 2 only. It will inhibit fatty acid transport into mitochondria. So the enzyme carnitine acyl transferase, it leads to the transport of long chain fatty acid. Um, it is a critical enzyme in the transport of long chain fatty acid into the mitochondria for beta oxidation. Here how it works. So this carnitine acyl uh, transferase or CPT1 is located on the outer mitochondrial membrane. It catalyzes the transfer of long chain fatty acyl group from fatty acyl coenzyme A to carnitine. It forms the fatty acyl carnitine. So this is an essential step for the transport of long chain fatty acid into the mitochondria. As fatty acyl coenzyme A, they cannot cross the inner mitochondrial membrane on its own. So basically, what this enzyme is doing, it is basically leading to the transfer of long chain fatty acid from outside to the mitochondria. So the correct option is, it will inhibit fatty acid transport into the mitochondria if the cell lacks the functional carnitine acyl transferase. I hope this question is clear. So let's move to our next question, question number 10. In a biosynthesis of histidine, the key intermediate that serves as the precursor for the imidazole ring is option number 1, 5, phosphoribosylamine. Second is 5, phosphoribosyl 1 pyrophosphate, PRPP. Third option is imidazole 4 acetate. Fourth option is urocanate. Please try to answer this question. We have two options, option number 2 and option number 3. Is there any other option correct apart from option number 3 and option number 2? Okay, so the correct option for this question is option number 2, PRPP. So let's see. So this biosynthesis of histidine, it is a very complex pathway which involves several steps, like 10 steps. This imidazole ring of the histidine is derived from 5 phosphoribosyl 1 pyrophosphate, which is key intermediate in nucleotide or amino acid biosynthesis. So what is the role of PRPP? It is a central precursor in the biosynthesis of histidine. It provides the ribose phosphate backbone, this backbone for the formation of the imidazole ring in the histidine. So, how the formation of the imidazole ring it happens? First step is this PRPP. It reacts with ATP. It forms the uh, phosphoribosyl ATP, and this phosphoribosyl ATP then it's converted through a series of step to this imidazole glycerol phosphate, so which contains the imidazole structure. So this the final step is this IgP, or the imidazole glycerol phosphate. It is further metabolized to form histidine. So this histidine pathway is has around 10 steps, which first this imidazole structure is formed, which ultimately lead to formation of the histidine. So the correct option for this question is PRPP, option number 2, 5-phosphoribosyl 1 pyrophosphate. I hope this question is clear. So if this question is clear, let's move to our next question, question number 11. Question number 11, it states that the enzyme involved in the conversion of aspartate to asparagine is, first option is asparagine synthetase, second option is aspartate transaminase, third option is asparated aminotransferase, fourth option is asparagine transaminase. Please try to answer this question. We have one option, option number 1. Is there any other option that might be correct apart from option number 1? So the correct option for this question is option number 1, asparagine synthetase. So this conversion of aspartate to asparagine, it is catalyzed by the enzyme asparagine synthetase. This reaction is involved in the addition of the amide group to aspartate, forming asparagine. So this group is being added to aspartate by asparagine synthesis, which is ultimately leading to the formation of asparagine. So the key point about this reaction are asparagine synthetase uses ATP and glutamine as a substrate, because this glutamine has NH3, so it uses ATP and the glutamine as the substrate. So basically what happens is we have aspartate, we have glutamine, and we have ATP. So in the presence of the enzyme asparagine synthesis, this reaction is catalyzed where the amide group is being transferred from glutamine to aspartate, which is leading to the formation of asparagine and glutamate and resulting in the formation of AMP and PPI. So the correct option for this question is option number 1, asparagine synthetase. I hope this question is clear. Okay, if this is clear, let's move to our next question. Question number 12. Uh, the role of pro-apoptotic protein like BAG and BIM is option number 1 inhibit caspase activity. Option number 2 promote mitochondrial permealization. Option number 3 activate DNA repair enzyme. Fourth option is prevent the formation of apoptosomes. Please try to answer this question. We have one option. Option number 1 inhibit caspase activity. So the correct, is there any other correct option for this question apart from option number 1? So the correct option for this question is option number 2. It promotes mitochondrial permeabilization. So let's see how. So these BAG and BIM, they are basically pro-apoptotic proteins, means uh, like the the proteins which are basically activating the apoptotic process. They are not directly involved in the apoptotic process, but they are involved in the initiation of the apoptotic process. Which, so the primary function of these protein is to promote the mitochondrial outer membrane permeabilization, which is leading to the release of the cytochrome C and other pro-apoptotic factor from the mitochondria to the cytosol where the apoptosis actually happen. So there are multiple steps how uh, uh, these these pro-apoptotic protein, they activate this or the promote this mitochondrial outer membrane permeabilization either by inhibiting anti-apoptotic proteins or by activating pore forming proteins. So basically we have cytochrome C in the mitochondria. We need this and other pro-apoptotic factors in the mitochondria for the apoptosis to happen, which occur in the cytosol of the cell. These proteins or these pro-apoptotic factors, they have to release from the mitochondria to the cytosol. So these protein, they leads to the this pore permealization of the mitochondria so that these proteins, they can go out into the cytosol and initiate the apoptosis process. So the correct option would be promote mitochondrial permeabilization. I hope this question is clear. So if this question is clear, let's move to our next question. Question number 13. The role of cytochrome C released from the mitochondria in the intrinsic pathway is: first, it activates caspase 8. Second, inhibit caspase activity. Third is forms the apoptosome and activate caspase 9. Fourth option is stimulates DNA repair enzyme. Please try to answer this question. We have one option. Option number 3 forms the apoptosome and activates caspase 9. So let's. The correct option for this question is option number C. It forms the apoptosome and activates caspase. So basically, when the cytochrome C gets released from mitochondria to cytosol, so it binds to APAF1, which is apoptotic protease activating factor 1, and ATP. This leads to the formation of apoptosome. This complex, which recruit and activates caspase 9, further which initiates the caspase process, which is ultimately leading to the apoptosis. So the correct option is it forms the apoptosome and activates caspase 9. I hope this question is clear. So if this is clear, let's move to our next question. Question number 14. The role of death receptor in the extrinsic pathway of apoptosis is: first, it inhibit apoptosis; second, activate caspase 3 directly; third, induce mitochondrial permeabilization; fourth, initiate signaling cascade leading to apoptosis. Please try to answer this question. We have one option, option number 3, induce mitochondrial permeabilization. We have another option, option number D. Are there any other option that might be correct apart from option number 3 and 4? The correct option for this question is option number 4. It initiates signaling cascade leading to apoptosis. So basically, death receptor are the cell surface receptor that plays critical role in extinction pathway of apoptosis. They are activated by specific extracellular ligands such as FAS ligand, TNF alpha, or TRAIL. So the key step for the extrinsic pathway is first the ligand binding, then the formation of death inducing signaling complex, further the activation of caspase 3, which is leading to the apoptosis process. So the role of these death receptor, it is initiating the signaling cascade leading to the apoptosis. So this uh, basically these death receptor, they are binding to the ligands which is forming these death inducing signaling complex. So it is initiating the signaling cascade leading to the apoptosis. So the correct option is option number 4th. So I hope this question is clear. So let's move to our next question, question number 15. Significance of BAX to the ratio BCL2 in apoptosis is, first option is it reflects the balance between pro-apoptotic and anti-apoptotic signals. Second option is indicates the ratio of DNA replication. Third option is it measures caspase activity. Fourth option is it quantifies mitochondrial permeability. Please try to answer this question. We have one option, option number 4. Is there any other option that might be correct apart from option number 4? So the correct option for this question is option number first. It reflects the balance between pro-apoptotic and anti-apoptotic signal. So let's see how we have BAX and BCL2. So if we go to this, so this BAX is pro-apoptotic protein while this BCL2 is an anti-apoptotic protein. So this BAX to BCL2 ratio is critical indicator of the cell susceptibility to apoptosis. So it basically, this ratio, it represents the balance between pro-apoptotic and anti-apoptotic protein in the cell, which determines whether the cell will undergo apoptosis or survive. So the key point about BAX and BCL2 are BAX is pro-apoptotic member of BCL2 family. It promotes the mitochondrial outer membrane permeabilization, which we have seen in earlier question, leading to the release of cytochrome C and activation of intrinsic pathway of apoptosis. The anti-apoptotic protein BCL2 is anti-apoptotic protein. This inhibits the mitochondrial outer membrane permeabilization by binding to and neutralizing pro-apoptotic protein like BAX and BAC. So basically these anti-apoptotic protein, they goes and bind to pro-apoptotic protein, so that they are not able to promote the permeabilization of the mitochondrial membrane. So it helps maintaining mitochondrial integrity and prevents apoptosis. So this Bags to BCL ratio, it indicates a high Bags to BCL ratio. It indicates a predominance of pro-apoptotic signals, making the cell more likely to undergo apoptosis. While the low Bags to BCL ratio, they indicates the predominance of anti-apoptotic signal, which is promoting the cell survival, not the cell um, death. So the correct option is option number 1. It reflects the balance between the pro-apoptotic and anti-apoptotic signal. So I hope this question is clear. So if this question is clear, this was our last question for today's session. So if you guys have any doubt in today's lecture, you guys can ask. So if there are no other questions, so this was end of our today's session. So thank you all for joining in today's lecture. So we'll meet next time at on Tuesday at 6 to 8 p.m. So we can end our session today here if you guys don't have any doubts. So if you guys have any doubt, you guys can ask, or else you guys can leave the meeting. Thank you all for joining in.
Ma'am, Tuesday next coming Tuesday.
Yes. Tuesday. My class is on every Tuesday from 6 to 8.
Okay, ma'am.
Okay.
Ma'am, can we get the presentation? Ma'am, is today what you have presented? Can we get that?